Now we're going to talk about the humoral immune response. So this is going to be B cells. So remember, B cells are still lymphocytes, which are white blood cells, and they are made in the bone marrow. And B cells, or B lymphocytes, stay in the bone marrow to mature. So what needs to happen is you have B lymphocytes that are made in the bone marrow and stay there to wait until they're fully mature and ready to meet their match. So you can think of it as they need to go through puberty. So they have to go stay in the bone marrow until they're ready, and they're ready to go out into the world and meet their match. So they have to get to the point where they're ready to meet their antigen. Remember, again, they're specific. So a B cell is going to look for its specific match. There's only one match for it. So one soulmate, you can look at it that way in the environment. So when it encounters its soulmate, its target antigen, then that stimulates what we call the humoral immune response. And when we look at humoral, this is going to be antibodies. So eventually antibodies, which are also specific to that one soulmate, to that match, are going to be produced. So again, antibodies are specific for the same. So a B cell that is meant to fight off the specific strain of flu is then going to make antibodies that will fight against that strain. So B cells are usually kind of what the captains of the ship, and then the antibodies are the workers. The antibodies are the ones that actually have the effect and fight against the antigen, which we'll see. So B cells lead to the secretion of antibodies that are dissolved in the blood. Again, B cells are not the ones that are producing the antibodies. They lead to what we call plasma cells, which you're going to see a little later. Plasma cells are the ones that make antibodies. So they're going to be from B cells. So you can think of it goes B cells, and we'll see there's memory cells too. Plasma cells make antibodies. So antibodies can circulate and fight against, again, that specific antigen that the B cell met. Most often, this is going to be bacteria and viruses, so pathogens that are in body fluid, meaning they're extracellular. We're going to look again at the T cells, which are cell-mediated, which is going to be intracellular pathogen, making sure we're covering both. Once antibodies are produced, then they can be circulating in the blood. Specifically, it's going to be in the plasma, part of the proteins in plasma, and also through lymph when it goes into the lymphatic system. There's tons of different kinds of B cells, 100 billion different kinds, not just 100 billion B cells, but different kinds of B cells. Because we're going to see that sometimes, say we have a virus that has a bunch of different kind of signals on its surface of its cell. Well, we're going to have B cells that may recognize just this part of the pathogen. We may have a B cell that recognizes just this part. B cell that recognizes this part. And a B cell that recognizes this part. So they're all fighting against the same pathogen. This is the same pathogen that has it all, but they're just recognizing different parts. So it just makes it so if you have a pathogen or an antigen inside your body, well, having ones that all recognize the same type and different kind of parts of it makes it more likely to find that match. So it's just increasing the likelihood that the immune system can recognize this pathogen that we're looking for. And then B cells are activated, so they meet their match, they meet their specific part on a pathogen or an antigen. then they are activated and a B cell will undergo differentiation. So we're going to make clones of ourselves, but then they're going to be making memory cells so they can remember later, and plasma cells to make antibodies. So antigens, again, are going to be recognized by certain B cells. So again, an antigen doesn't necessarily need to be the entire cell or entire virus or bacteria. It can be a piece. So it can be one little section is considered an antigen. So technically, this could be an antigen or this could be an antigen, because either way, we're going to have a B cell that recognizes it and cause a reaction. So antigen is still anything that causes an immune response. It doesn't necessarily need to be the entire virus or the entire bacteria. It just needs to be some part of it that's going to cause an immune response. B cells, say they have a receptor, this B cell has a receptor for this specific kind of part of a pathogen. What happens is it recognizes its specific match. So these are matches. This is what the B cell's made for. Then we have what we call receptor-mediated endocytosis of that antigen receptor complex. So what's going to happen is that B cell is going to take the receptor and its antigen inside its cell. This is when we get activation. So you find your antigen and then you bring it inside the cell. Remember, B cells also are going to process this antigen because they have MHC2. So remember, they're antigen presenting cells too. So then they're going to, on their MHC2, they're going to present piece of that antigen as well. So not only are they going to process it and present it, this is going to lead to its activation, where it's going to be proliferation, meaning it's going to make more clones of itself. It's going to start dividing and make a clone army. And then it's going to also lead to differentiation into effector cells, which are going to be our plasma cells and our memory cells. So again, clone army, then the clones are going to go to plasma cells and memory cells, and plasma cells are going to make the antibodies. So most of our clone cells, once we start to proliferate, we just make a big copy of clone army again, are going to become those plasma cells. Plasma cells are the ones that are producing and secreting antibodies. So then the antibodies are going to be what fights against the antigen. And usually what we say is they mark the antigen for destruction by a phagocyte. Most often it's going to be macrophages. So what happens, and we'll look at the different types of antibodies, they're going to say, again, we have a pathogen that's coming around. The antibodies will surround it in different ways, and we'll talk about those ways later. But it's going to surround it and say, okay, this needs to be killed. So it's going to neutralize it so it can't have its effect. And it's going to say, okay, now we need something to come along and eat it. Most likely, again, it's going to be macrophage that comes along, is going to be our Pac-Man, and it's going to eat this and saying, okay, that's something I need to eat. And again, macrophages are part of it, but they would eat anything. So they'll eat something that this is covered in antibodies, so it would eat that. So that's again why it's not specific, because it's going to eat anything that you tell it to. So again, clone cells. So we proliferate. So B cells are activated. They make a bunch of themselves. And if they're not made into plasma cells, then they're made into what we call memory cells. This is so we can remember. Again, so this will be if you meet the pathogen, so whatever pathogen's there, if you see that person again, you'll be able to recognize it a lot faster. Again, it's like studying. So you learn this the first time and you're like, okay, I understand. And then you go back to it and you repeat it. And so you have a faster reaction and answering questions about it the more times you see it because you have more memory cells. Think of it that way. It's not exactly, but since you have more memory cells, the more likelihood you're going to remember that same antigen in the future. So just better picture. So this is looking at a B cell and it has its specific receptors looking for its antigen. Oftentimes, again, it's extracellular. So B cells are exposed to its antigen and the antigen binds to the receptor. So you can see the antigen and then the receptor on the B lymphocyte. If it was this B lymphocyte, it's not a perfect match. So this antigen is not going to be able to bind to that receptor. It's only to this guy over here and won't be able to bind to this receptor because, again, it's only this guy in the middle. The B cell, and we're going to talk about helper T cells when we get to them. This, again, the B cell is an antigen presenting cell. So it is going to take in these receptor complexes and it's going to process the antigen and put it on its MHC2. And that's going to communicate with helper T cells, which again are the party planners. So helper T cells are going to coordinate everything. They're the major coordination cell. So this is going to help kind of say bring help and reinforcements to the B cell that is fighting off its antigen. So it's just making sure there's more communication that the B cell can have some backup. Then we go to clonal selection. So again, you're making copies of itself. So mitosis is going to make multiple copies. And then it's going to differentiate. Again, plasma cells are our effector cells. So it's going to be mostly plasma cells that will make antibodies. And then these antibodies will start fighting against the antigens. And then we have one that's a memory cell here that it's going off and it's waiting until, or if we are exposed to the same antigen again, it's ready to fight as soon as possible. So we don't need as many steps.